So, dear students, after we have finished the study of the male reproductive world, that is the androecium, now let us begin our discussion with the female reproductive structure. And the female reproductive structure, as you know, is the gynoecium. So, here, gynoecium is the female reproductive world, and the individual unit, the single unit of the gynoecium, is called as a pistil or a carpel. So, pistil or carpel is a term which is used for the single unit that makes up the gynoecium. In gymnosperms, we see the presence of the megasporophyll. Fill matlab leaf. Megasporophyll matlab a leaf which can produce megaspores, the female spores. The same job is done in angiosperms by the pistil or the carpel. So, in a way, for MCQs, you'll remember that the pistil or the carpel is a modified megasporophyll, and the carpel is divided into three parts. That is the stigma, style, and the ovary. And the stigma is a landing platform for the pollen grains. So during pollination, the pollen grains first fall on the stigma. Then the pollen grain results in the formation of a pollen tube. The pollen tube passes through the style in order to enter in the ovarian cavity. From there, it will enter into the ovule. So this is the landing place. Then there will be formation of a pollen tube, ultimately to reach the ovule, followed by double fertilization. And after fertilization, the ovary will change into a fruit and the ovule will change into a seed. So it is the female gynoecium, or rather the carpel, and the ovary and the ovule part of the carpel, which are to be carried forward in the next generation. Okay, so just like a human female in her uterus carries the fetus, the embryo, that is the future generation, which is given food by the endosperm, is carried by the ovule and the ovary of the carpel, which is a part of the gynoecium. So the pistil, if it is a single pistil, it is called monocarpellary. Many, it is called polycarpellary. And we have studied about syncarpus and apocarpus conditions in morphology of flowering plants. So I don't find it the need to repeat it again over here. The ovule is defined as a integumented megasporangium. Why integumented megasporangium? Let us study shortly. In a typical ovule, we see the following parts. There is a central diploid mass of parenchymatous cells, which is called as the nucellus. This nucellus is actually a nutritive tissue, and it is also called as the megasporangium proper, and it is surrounded by two protective coats, which are called outer integument and inner integument. So that is why the ovule is an integumented megasporangium. In the future, this outer integument will form the testa of the seed coat, and the inner integument will form the tegmen of the seed coat. Now, if you observe this diagram, the integuments do not completely cover the nucellus. They always leave an opening at the top, which is called as the micropyle. This opening is called as the micropyle. And the stalk of the ovule is called funicle. The point of attachment of the funicle to the body is called as the hilum. So you have the stigma, style, ovary, nutritive tissue, placenta, ovules. This point of attachment is called as the hilum. Stalk is called as the funicle. Rest of the part that we discuss is considered as the body of the ovule. The base of the ovule is called chalaza. Look at this diagram that I'm drawing. Here, the micropyle, chalaza, and the funicle are in a straight line. So it is called as a orthotropous ovule. Whereas in this particular ovule, where the micropyle and the chalaza, base of the ovule, are in one line, but the funicle is not in line with them, is called as the anatropous ovule. And majority of the ovule in angiosperms are anatropous. Okay. Now here, further. So in the ovule, one of the important parts is the embryo sac. If you look at a typical embryo sac, then it shows the presence of the following structures within it. And the embryo sac that I'm going to discuss with you is called as the polygonum type. And this polygonum type is 7-celled, 8 nucleated type, where the embryo sac shows the presence of 3 cells, which are called as the antipodals. They are haploid towards the chalazal end. In the center, there is a large cell called central cell. It is binucleate, showing the presence of 2 polar nuclei. Towards the micropylar end, I see the presence of an egg with 2 cells, which are called synergids. The egg and the synergid together is called as the filiform apparatus. I'm sorry, the egg and the synergid together is called as the egg apparatus. The synergids show the presence of a filiform apparatus, which helps in the pollen tube entry into the embryo sac. So if you count the number of cells, one, if you count the number of cells, let us count the number of cells. 1, 2, 3, 4, 5, 6, 7. And each cell has a single nucleus except the central nuclei. So that is why you have 7-celled, 8 nucleated structure, which is the polygonum type of the embryo sac. It was observed in a species called Polygonum species. So how does this embryo sac develop? Okay, the embryo sac. So how does the embryo sac develop? The embryo sac develops by a process called megasporogenesis. It develops by a process called megasporogenesis, followed by megagametogenesis. Okay, so let us quickly see these two steps of megasporogenesis and the megagametogenesis. So, one of the diploid cell of the nucellus becomes a megaspore mother cell. And this megaspore mother cell undergoes a reduction division, which is called meiosis or megasporogenesis. Results in the formation of four haploid megaspores, out of which three degenerate, and one haploid megaspore is called as the functional megaspore. Now, this functional megaspore, the wall increases, the cell elongates, and this wall of the functional megaspore is the future wall of the embryo sac. It has a single nucleus which undergoes first mitosis, and now it results in the formation of two nuclei, one towards the micropylar end, one towards the chalazar end. Now it undergoes second mitosis to form four nuclei, two towards micropylar end, two towards the chalazar end, which undergo third mitosis, resulting in the formation of eight nuclei, four towards the micropylar end and four towards the chalazal end, four towards the micropylar end and four towards the chalazal end. One nuclei each from the micropylar and the chalazal end migrate towards the center, and they will result in the formation of the polar nuclei. The other three towards the micropylar end will form the egg apparatus. The remaining three towards the chalazal end will result in the formation of the antipodals. Thus, these three mitoses complete the process of megagametogenesis, and we will get the formation of a mature embryo sac. So that is how the development of the embryo sac occurs. It is a monosporic type of development. It is a monosporic type of development because it involves the use of only one functional megaspore. So such a development is called as monosporic. It occurs inside the sporophyte, that is the ovule. That is why it is also called as monosporic endosporic type of development. So this type is called as monosporic endosporic type of development.